There is a kind of blood that, under the wrong circumstances, can turn a mother's own body against the child she's carrying. By every rule evolution obeys, the people who carry it should have vanished from the earth a long time ago. Their bloodlines should have thinned with each generation until there was no one left. That isn't what happened. Tonight, roughly 1 in 7 people alive are carrying it, and it may be moving silently through you as you lie there listening without your ever having felt a thing. It has a dull, ordinary name, the kind you might have signed on a form without a second thought, being RHD negative. It's the negative in a blood type, the missing half of positive or negative. About 15% of people have it, though that figure swings wildly depending on where on earth you look, from almost no one in some places to nearly a third in others. And here is the part that should keep you awake a little longer than you meant to be. For close to 90 years since the season it was discovered, the most basic question about this blood has had no answer. This isn't a question someone invented to sell you a theory. It's a real one, argued over in real scientific journals, by people with no patience for wonder and every interest in being right. Why is it still here at all? Because the cost of it is almost too cruel to say plainly. An RHD negative mother can carry her first baby perfectly safely, and then, without any warning anyone could have read, lose the second, or the third. The body built across millions of years to be the safest place a child will ever know, tricked into treating that child as an intruder. A trait that quietly takes your later children is the precise kind of thing nature deletes completely, and fast. This one refused to go, so we are left with a riddle that has outlived the scientists who first noticed it, and their students, and the students who came after them. We can find this blood in anyone, map it across the whole human species, and read the exact letters of code that have gone missing. We can even stop the harm it once did. And we still cannot tell you why natural selection never swept it away. That's what we're going to sit with tonight, in the dark, together. And there's something quietly rare about tonight in particular. Because this is uncommon blood, because it may be the very thing in your veins right now. You aren't only hearing this story, some of you are it. Scattered across the world at this exact moment, other people are resting the way you're resting, carrying the same small absence. One of them where the sky is already turning grey with morning, another in a silent house long past midnight, and one more in the green mountains of northern Spain, where this whole story reaches its strange peak, where this blood runs thicker than anywhere else alive. The same absence, the same open question, all of them inside the same handful of hours as you. If somewhere in the next couple of hours that thought comes back to you, the wondering of whether you might be one of them, hold on to it. Don't do anything with it yet. There's a small, easy, quiet way to find the others, and I'll come back to it right at the very end. And if you came here to fall asleep, you can let your eyes close now. You don't have to follow every word. Let it drift past you like weather. We're going to follow this blood everywhere it goes. Back to a laboratory full of monkey blood on the edge of the Second World War, and up into a mountain people whose language is related to no other tongue on earth. We'll look hard at the wild stories that have been hung on it, the lost continents and ancient bloodlines, the secret powers it supposedly grants, and watch which ones fall apart the moment you touch them with evidence. And the whole thing arrives, slowly, at the real edge of what anyone knows, the place where the scientists themselves go quiet. So let's begin where the strangeness begins. Before the missing piece of code, there's the thing it goes missing from. A single drop of blood. Take a single drop of your blood. Less than that, really. A smear too small to see clearly. Inside it are millions of red blood cells, and each one is a small marvel we tend never to think about. They are shaped like little discs, pressed in at the centre, almost like a sweet pinched on both sides, and that shape is not an accident. It gives each cell the most surface for its size, so it can soak up oxygen in your lungs and let it go again out in your tissues, over and over, hundreds of thousands of times in a single cell's short life. They have no nucleus, these cells. They throw it away as they mature like a traveller shedding luggage, to make room for more of the cargo they exist to carry. They are in a sense the most single-minded cells in your body, born to carry, nothing else. But on the outside of each of these little carriers, studding the surface like markings on a ship's hull, are proteins and sugars, thousands of kinds of them. And some of these surface markings are the reason blood comes in types at all. This is the part the whole night turns on. Your immune system is, at heart, a machine for telling self from not self. It spends your entire life walking the corridors of your body checking identification. The cells that belong to you wear the right markings, the right flags, and the immune system waves them through. Anything wearing the wrong flags or no flags it recognizes, it attacks. That is, most of the time, exactly what you want it to do. It is how you survive a world full of things that would happily make a meal of you. The surface markings that matter for blood typing are called antigens, and an antigen is just, in the end, a flag the immune system can read. The most famous of these flags make up the ABO system, whether your red cells fly the A flag, the B flag, both, or neither, which is what gives us blood types A, B, AB, and O. This is the part most people half remember. If you pour blood carrying a flag your body has never seen into someone whose immune system doesn't recognize it, that immune system can react violently, clumping the cells, breaking them apart, sometimes killing the person. This is why blood has to be matched. It is also why, in the early days of transfusion, before anyone understood any of this, people sometimes died on the table for reasons no one could explain. The blood was right there, red and warm and alive. It just wore the wrong flag. Layer on a second set of flags, and this is where our story actually lives. On top of the A and B system, there's another, entirely separate one. It's called the RH system, and it is far larger and far more complicated than most people realize. More than 50 distinct flags, all riding on the same family of proteins. But one of those flags matters more than all the others put together, because it is the strongest, the most likely to provoke a reaction. It's called the D antigen, and the entire positive and negative on your blood type comes down to this single flag. If your red cells fly the D flag, you are RHD positive. That is most of humanity. If your red cells do not fly the D flag, if that one marking is simply absent, then you are RHD negative. That is the 1 in 7. That is, possibly you. And notice how quiet the difference is. An RHD negative person is not sick. They are not weaker or different to look at or set apart in any way you could ever notice from the outside. They are missing one flag out of thousands on the surface of one kind of cell. In ordinary life, it changes nothing. You could go your entire existence and never know, and most people through most of history did exactly that. The absence only ever announces itself in two situations: when your blood meets someone else's in a transfusion, and when a mother's blood meets her baby's. We'll come to both. But hold on to how small this is. A single missing flag. That is the whole physical reality at the centre of every wild story you have ever heard about negative blood. One protein, present in most of us, gone in some of us. The natural question, the one that should be forming in the back of your mind as you lie there, is this: how does a flag just go missing? And the answer is the first genuinely strange thing in this story. In most people of European descent who are RHD negative, the flag isn't broken or hidden or switched off. The instructions for building it are simply not there. The gene that would build the D antigen, a gene called RHD, sitting on the short arm of chromosome 1, has been deleted. Cut clean out of the genome, both copies, the one from your mother and the one from your father. Where a positive person has a working gene, a negative person of European ancestry, in most cases, has a gap, a silence, nothing to read. That is not a small mutation. A single letter of DNA changed, a typo, happens constantly, all the time, in all of us, and most of the time it means nothing. But the wholesale deletion of an entire working gene, inherited in one piece, carried forward intact through tens of thousands of years and untold generations until it arrives. Tonight, in 1 in 7 of us, that is a different order of thing. That is not noise. It is a signal. The question is what it's signalling. And here is the first hint that this is going to be stranger than it looks. The way this gene got deleted, the precise mechanism, has to do with the unusual way these genes are arranged in the genome. The gene for the D flag and its near twin, the gene next door that makes the other arch flags, sit close together, facing in opposite directions, flanked by nearly identical stretches of DNA, repeated sequences that scientists have nicknamed the rhesus boxes. And when DNA copies itself, regions like that, where the same sequence repeats, are exactly where the machinery slips, where it lines up the wrong repeat with the wrong repeat, and in the slip loops out the gene between them and snips it away. The deletion that defines 15% of humanity wasn't a random act of damage. It was, in a sense, a structural accident waiting to happen, built into the architecture of that little piece of chromosome. an accident the genome was, in a way, always set up to make, so it happened. Somewhere in someone deep in our prehistory the loop slipped and the gene was gone. That person had children, and the gap was passed on. And here is where the real story begins. To understand why that gap survived, and where it travelled, and why it should have been a dead end and wasn't, we have to leave the body entirely and go and look at a laboratory in New York, on the edge of the Second World War, full of the blood of monkeys. The year is 1939, sliding into 1940. The place is New York, and the man at the centre of it is one of the quiet giants of medicine, a man named Karl Landsteiner. You may not know his name, but you have almost certainly benefited from his work. Decades earlier, back at the turn of the century, Landsteiner had been the one to figure out the ABO blood groups, the A, B, AB and O flags we just talked about. Before him, transfusion was a kind of lottery. After him, it became something a doctor could do on purpose, safely, knowing why. For that, he had won the Nobel Prize. He was not a man looking for wonder. He was a careful, methodical scientist, and in 1939 he was doing what careful scientists do, a routine experiment, expecting a routine result. Working with his colleague Alexander Wiener, Landsteiner took blood from rhesus macaque monkeys, small sharp-faced monkeys common in laboratories, and injected it into rabbits and guinea pigs. The animals' immune systems did what immune systems do. They saw the foreign monkey blood flags, didn't recognise them, and built antibodies against them. Antibodies are the immune system's wanted posters, molecules shaped to lock onto one specific flag and mark it for destruction. So far, nothing surprising. Then came the step that made history almost by accident. The researchers took those anti-monkey blood antibodies and tested them against human blood just to see. And the human blood sorted itself into two camps. In about 85% of people, the antibodies latched on. Those people's red cells were carrying a flag that looked, to the antibody, like the monkey's flag. But in about 15% of people, nothing happened at all. The antibodies drifted past those red cells as if they were invisible, as if something every other person carried was, in these people, simply not there. They had stumbled onto the D flag and onto the people who don't fly it. They named what they'd found the arc factor, or rhesus, after the monkey whose blood started it all. And here is a small, honest footnote that I love, because it tells you something true about how science actually works. The human flag and the monkey flag, the thing they thought they were detecting, later turned out not to be the same thing at all. They were similar enough to fool the early test, but they were different molecules. By the time anyone sorted that out, the name rhesus was already everywhere, in every textbook, on every form, so it stayed. To this day, the most important blood group system after ABO is named after a monkey antigen that it isn't really the same as. The world is full of names like that, fossils of old mistakes, carried forward because changing them would be more trouble than living with them. There's something almost comforting in it. Even our most precise sciences are wearing, here and there, the scars of their own first guesses. But the discovery did something far more important than name a thing. It solved a horror that doctors had been documenting helplessly for generations. For as long as there had been midwives and physicians keeping any kind of record, there had been a particular terrible pattern that no one could explain. A woman would have a baby, a healthy baby, pink and crying and fine, and then, a few years later, she would have another, and this one would be born wrong, yellow, deeply jaundiced, swollen with fluid, struggling to breathe. Sometimes it would die within hours, sometimes within days, and then it would happen again with the next. The first child healthy, the later children doomed, always in that order. Whole families in some lineages lost child after child this way, and there was no understanding of it to be had, only grief, and the search for an explanation in the only places people had to look. Bad luck, bad air, a curse, the will of God, or something the mother had done or failed to do. Imagine carrying that. The second loss, then the third, and being told by everyone you could ask that there was simply nothing to be done and no reason it was happening. Right around this time, across 1939 into the early 1940s, a physician named Philip Levine pieced the pattern together and tied it to the newly described arch factor, and the whole grim machinery suddenly made sense. Here is what is happening, and we'll go slowly, because this is the beating heart of the whole story. Take a mother who is RHD negative. She does not fly the D flag. She is carrying a baby who is RHD positive, who does fly it, inherited from the father. Through the pregnancy, mother and baby keep their blood mostly separate. That's how it's designed. But at the birth, and sometimes a little before, some of the baby's blood crosses into the mother, and the mother's immune system, that lifelong machine for telling self from not self, encounters the baby's D-flag, a flag it has never seen, a flag it does not carry, a flag that reads, to it, as foreign, as intruder. With the first baby, this usually causes no harm at all. The immune system needs time. It sees the foreign flag and it begins, slowly, to build its wanted posters, its antibodies against D. But that first baby is usually already born and safe by the time the response is fully built. The mother walks away with a healthy child and a body that has now, quietly, learned something. It has made antibodies against the D flag, and, this is the cruel part, it has remembered how. The immune system never forgets a face, the antibodies wait. And then comes the next pregnancy, another Rhd positive baby, flying the same D flag. But this time the mother's body is not naive, this time it is armed. Those waiting antibodies, and crucially, antibodies of a kind small enough to cross the barrier into the baby, pass into the baby's bloodstream and find the baby's red cells, studded with the very flag they were built to destroy. And they do what they were built to do. They mark the baby's red blood cells for destruction, and the baby's own body, doing its job, tears them apart. The baby becomes anemic, starved of the cells that carry its oxygen. The breakdown of all those cells floods its system with a yellow pigment, which is the jaundice. In the worst cases, the baby's tissues swell with fluid, its organs strain, and it can die in the womb or shortly after birth. The disease has a clinical name now, hemolytic disease of the fetus and newborn, and its most severe, swollen form carried an older Latin name in the medical texts, hydrops fetalis. But to the families who lived through it, century after century, it needed no name at all. It was simply the babies who didn't live. And once Levine had laid this out, the old pattern made perfect, awful sense. First baby fine because the mother wasn't yet armed. Later babies in danger because now she was. A mother's body built across millions of years of evolution to be the safest place in the world for a child, turned, by a single missing flag and the immune system's long memory, into the most dangerous. This is the first real clue to the mystery and the moment to let it land. Because the instant you understand this, the instant you really feel it, you understand the paradox at the center of everything. A trait that does this should not survive. Think it through with the cold logic of evolution. Every later child it kills is a child who never grows up to carry the trait forward. The gene that causes the harm is, in effect, deleting itself from the future every time it does its harm. Over enough generations, a trait that cost you your children should be pushed steadily downward, toward the vanishing point. That isn't mysticism. It's close to what the basic arithmetic of inheritance predicts. And as we'll see, the real picture carries a crucial wrinkle that the simple version misses, one that only deepens the puzzle rather than dissolving it. And yet, when scientists began, in the decades after Landsteiner, to map this trait across the whole human species, to count, population by population, who flew the D flag and who didn't, they did not find a trait limping toward extinction. They found one that had not just survived. In places it had flourished. In one place it had very nearly won. And the map they drew is so strange, so sharp, so suggestive of something we cannot quite read, that it has fed 100 years of theories, true and false alike. Let's go and look at it. If you could lift up off the surface of the earth and look down at humanity not as nations or cultures, but as this one single trait, colouring in every region by how common Rhd negative blood is there, you would not see an even wash. You would not see 15% smeared smoothly across the globe. You would see something with a shape, a pattern with an obvious centre and an obvious direction, the way iron filings arrange themselves around a magnet. And the moment you saw it, you would want to know what the magnet was. The centre, the epicentre, is a small region in the mountains between Spain and France, along the western end of the Pyrenees, on the edge of the Atlantic. It is the homeland of the Basque people. And among the Basque, Rhd negative blood reaches the highest concentration found anywhere on Earth. And the strangest part is how we even came to know it. The man who first noticed wasn't working in those mountains. He was on the far side of the world in Argentina. His name was Miguel Angel Echeverri, a hematologist born on the Argentine plains in 1905, the son of Basque immigrants. His mother had come from a village in Gipuzkoa, his father's people from Lower Navarre, part of the great wave of Basques who crossed the Atlantic in the late 1800s and early 1900s. So many of them went to Argentina that today around 1 in 10 Argentines carries some Basque ancestry. A local paper later described families like his with a phrase I can't improve on: people who planted wheat and harvested doctors. In the 1940s, this doctor, 2 generations removed from those valleys, began studying the blood of the Basque families around him, his own transplanted people, and he found a frequency of Rhd negative blood unlike anything on record. In one group of Basque Argentines, more than a third came back negative. So the most Basque blood on earth, in a sense, first showed itself thousands of miles from home in the veins of emigrants who had carried it across an ocean without ever knowing it was in them. The mountains had sent their strangeness abroad, and a grandson of those mountains read it back to them. The numbers around all this are exactly where the internet versions fall apart, so let's get them right, because that's the whole reason a telling like this earns your trust. You'll hear figures like 40 or 45% thrown around, and they usually come from mixing up 2 different things that both happen to be measured in percentages. One of them is just how many Basque people actually are Rhd negative, the ones who, like Echeverry's donors, would test negative on the spot. That sits around a third, somewhere near 30 to 35 in every 100. Already the highest in the world by a wide margin. Across most of the planet is well under 10 in 100, and in many places almost none. The other figure climbs higher, and it's the one that gets misquoted, and it's worth slowing down for, because it's also the quiet key to the mystery waiting at the end of tonight. You carry 2 copies of this gene, one from each parent. The question here isn't how many people show the trait, it's how many copies of the deleted version are drifting through the whole gene pool, the hidden ones included. In 2018, a team sequenced the DNA of dozens of Basque volunteers, people chosen because all 4 of their grandparents had been born in the same valleys, roots running straight down into the local ground. When the gene copies were counted, nearly half came back as the deletion, around 47 in every 100. Both numbers are true, they answer different questions, and the gap between them hides the most important character in this whole story. To actually be Rhd negative, you need the deleted copy from both parents. So take a Basque couple, both positive, both flying the D flag, neither with any reason to suspect they carry anything out of the ordinary. Each is quietly holding one deleted copy underneath. On average, 1 in 4 of their children will inherit the deletion from both sides and be born Rhd negative, seemingly from nowhere, into a family that believed it had no such thing. That child isn't an accident in the bloodline. It's the moment a thread that had been moving invisibly through the family for who knows how many generations finally steps into the light. Hold on to those hidden carriers, the positive testing people with 1 deleted copy tucked away and no idea it's there. Much later tonight, they turn out to be the answer to almost everything, so the headline survives all the misquoting intact. The Basque country is the world's peak, the magnet's centre, and the Basque are strange in more than their blood. This is the part that makes the hair stand up a little, the part that the wilder theories absolutely cannot resist. Because the Basque are also a linguistic island. Their language, which they call Euskara, is unlike any other on Earth. They don't even name themselves by blood or border. They call themselves Euskaldunak, which means simply the ones who speak Basque. It is not part of the great Indo-European family that includes English and Spanish and Russian and Hindi and nearly every other tongue spoken from Iceland or India. It is not related to Finnish or Hungarian, the usual European outliers. It is not related to anything. Linguists have searched for a living relative for it for as long as there have been linguists, and they have found none. It stands alone. While wave after wave of migration and conquest rolled across Europe over the millennia, each one carrying its own language and overwriting what came before, while the languages of whole peoples vanished into the languages of newcomers, Euskara held. It survived as an island of older sound in a sea of incomers. So you have a population that is an island in 2 completely separate ways, an island in its language, holding on to something ancient that exists nowhere else, and an island in its blood, holding on to a genetic trait at a frequency found nowhere else. The same people, set apart on 2 measures that have nothing to do with each other. You can feel, can't you, why this is irresistible to the storytellers, why the temptation is to say the Basque are something special, something left over, the last of an older kind of people. We are going to take that temptation seriously, and then we're going to see honestly how much of it the evidence actually supports. But first, let's finish the map, because the rest of it matters just as much. Move outward from the Pyrenees, and the frequency of Rhd negative blood doesn't just drop off a cliff. It fades in an orderly way, like ripples spreading from where the stone went in. Stay along the Atlantic edge of Europe, Ireland, Scotland, the western fringes, Brittany on the French coast, and it remains high, around a quarter of the population. These are the so-called Celtic fringes, the western edges, and they hold on to a lot of it. Then, as you move east across the body of Europe, it falls. It thins through Central Europe, drops further around the Mediterranean, and is thinner still by the time you reach the Middle East. Out among the great populations of East Asia, it has all but disappeared. In many parts of China and Japan, Rhd negative blood is found in well under 1% of people. A trait that touches a third of one population touches almost no one in another. And then there's a second cluster that the wild theories love, because at first glance it seems to break the simple picture. Across the water from the Basque, in the mountains of North Africa, among the Berber peoples of the Rif and Kabylia, in what is now Morocco and Algeria, Rhd negative blood appears again at 15 to 20%. High, far higher than the surrounding regions. A second peak separated from the first by the Mediterranean Sea. A sensational version of the story would stop there and make this a mystery. Two isolated peaks, on two continents, surely some hidden connection, some lost people who once spanned the sea. But here the truth is better than the mystery, because we can actually read the genes, and when geneticists looked at the Berber Rhd negative blood, they found that it is caused by the very same deletion, the same gene cut out in the same way as in the Basque and other Europeans. Not a similar trait that happened to arise twice, the same one, which tells us these are not two separate accidents. They are two ends of one thread, a genetic link written into the DNA connecting the mountains of northern Spain to the mountains of North Africa. The likeliest explanation is old human movement, people carrying the deletion crossing between Iberia and North Africa in deep prehistory, leaving the same signature on both shores. No lost continent required, just people and boats and time. And here is the most important honesty of the whole map, the place where we most sharply part company with the sensational versions. You will hear, again and again, that Rhd negative blood is absent from Africa, absent from East Asia, absent from the indigenous peoples of the Americas and Australia, and that this absence is a clue, even a sign of some separate origin for the people who have it. It is not quite true, and the way it's untrue is genuinely interesting. Take Africa. It's true that the European-style deletion, the clean cutting out of the Rhd gene, is rare in most of sub-Saharan Africa. But Rhd negative blood itself does occur there. It just gets there by a different genetic road. In many Africans who don't fly the D flag, the gene hasn't been deleted at all. Instead, it's present but broken, a version scientists call a pseudogene, carrying a small duplicated chunk and a stop signal in the wrong place, so that the cell starts to read the gene and then halts partway, building nothing usable. The flag never gets made, even though the instructions are technically still there, jammed and unreadable. In other people, there's a hybrid gene, a mosaic of the D gene and its neighbour stitched together, that also fails to produce the flag. So the destination is the same, no D flag, Rhd negative blood, but the roads there are completely different. Europeans have it and Africans don't is simply wrong. It would be more accurate to say that humans have found several different ways to lose the same flag, and different populations lost it differently. That detail matters enormously, and we'll come back to it, because it's one of the strongest clues we have about where all this really started. So what is the magnet? What explains the shape of the map, the sharp Atlantic peak, the orderly fade to the east, the near vanishing in the Pacific world? The mainstream answer, the one most population geneticists would give you, is not exotic at all. It is the slow churning of human history. Over the last 10,000 years or so, Europe was not a sealed room. It was crossed and recrossed by great migrations, farmers spreading out of the Near East, then later waves of peoples coming in from the steppes of the East, each bringing their own genes and mixing with, and partly replacing, the people already there. Each of those incoming waves carried mostly Rhd positive blood, and as they swept across the continent mixing in, they diluted whatever was there before. But they didn't dilute it evenly. The places hardest to reach, the places at the far western edge with the sea at their backs, the Basque country, the mountains of the Atlantic fringe were the least disturbed. The waves reached them last and weakest, and so those places preserved more of the older genetic makeup of Europe, including a higher frequency of the Rhd negative deletion, while everywhere else it got watered down by the incomers. If that's right, then the Basque are not special because they invented this blood, or because they're a different kind of people. They may be special for a much more poignant reason. Because they kept it. While it was being diluted everywhere else, they held on. Their mountains and the strange durability of their language are part of the same story. A population at the edge of the map, sheltered just enough, holding on to older things long after the rest of the continent had let them go. A kind of living museum standing at the western wall of Europe with the sea behind it. That explanation is good. It is probably largely true. And this is the crucial thing. It does not touch the real mystery at all. Because migration and mixing can explain how a trait got spread around, concentrated here and diluted there. It is a perfectly good account of distribution. But it says nothing about the question that started us off. It does not explain why natural selection didn't simply delete this trait everywhere, given what it does to later pregnancies. A trait being old and preserved at the edge of Europe is not the same as a trait being safe to carry. The cost is still there. The downward pressure is still there. Migration tells you where the trait is. It does not tell you why the trait is allowed to be anywhere at all. For that, the map isn't enough. We have to ask where this blood truly came from, not which mountains preserved it, but in whom and when the gene first slipped away. And that question takes us much, much further back than the Basque or Europe or even our own species. Before we chase where the deletion came from, it's worth slowing down on something that almost every version of this story skips entirely, and skipping it is how people get fooled. The question is simple. What is the D flag actually for? What does this protein, present in most of us and missing in some of us, actually do in the body? And the honest, slightly unsettling answer is that we are still not completely sure. Let's start with what we do know. The D flag is one member of a family of related proteins that sit in the membrane of the red blood cell, threaded through it like a stitch passing in and out of fabric, 12 times in fact, weaving back and forth across the cell wall. Proteins shaped like that, threaded through a membrane that many times are almost always doing one particular kind of job. They are channels, gates. They let specific small things pass through the cell wall that otherwise couldn't easily cross it, and the best evidence we have says that this family of proteins is a gas channel. The leading candidates for what it transports are ammonia, the same sharp-smelling compound your body has to deal with as a waste product of breaking down proteins, and possibly carbon dioxide, the gas you breathe out. The idea is that these proteins help shuttle these molecules across the red cell's membrane, part of the constant invisible chemistry of keeping your blood balanced, your waste moving, your acidity in range. Some of the proteins in this family are clearly the workhorses of that transport. The D flag and its close cousins may be more like structural partners, holding the working channel in place, anchoring it to the cell's skeleton, rather than doing the heavy lifting themselves. But, and this is the part to sit with, the precise job of the D protein specifically, the very flag that 15% of us are missing, is still not nailed down. We know people can live perfectly normal lives without it. That much is obvious, since 1 in 7 of us do. We know that people who are missing the entire family of these proteins, a vanishingly rare condition we'll meet near the end of the night, do have real problems, fragile misshapen red cells and mild anemia, which tells us the family as a whole matters, but the role of the single D flag, in isolation, still partly open. So even before we get to the great evolutionary riddle, there's a smaller mystery nested inside it. We're talking about a protein whose exact purpose we cannot fully state, missing from a seventh of humanity for reasons we cannot fully explain. The unknowns go all the way down. Here is the part that genuinely reframes the whole story, and that the sensational versions get exactly backwards. They love to tell you that Rhd negative blood is somehow alien, or non-human, or a sign that its carriers came from somewhere else, that the Rh factor is the marker of an outsider lineage. What's actually true is almost the precise opposite. This protein family is not alien. It is one of the oldest and most deeply shared pieces of machinery in all of life. Follow the family tree back and it gets staggering. The ancestor of these gas channel proteins doesn't just appear in humans or in primates, or even in mammals. Versions of it show up across the entire living world. They're in fish, in the gills of fish. where they help dump ammonia straight into the water. They're in worms. They're in the tissues of sponges, among the simplest animals there are. They're even in bacteria and single-celled organisms, the deepest, oldest branches of life on Earth. This is plumbing so fundamental, so useful, that life invented it almost at the beginning, and has been keeping it, in one form or another, ever since. When did the particular branch that became our arc protein split off from its even more ancient relatives? That reaches very far back, long before there was anything you would call an animal, with a backbone walking on land, deep in the history of the cell. Try to hold that for a moment in the dark. The deep root of the thing that gives you a positive or negative blood type was already present, doing related work, in living things so far back that calling them our ancestors is almost too gentle a word. The more recent split, the one that produced the specific pairing of genes we carry, the D-flag gene and its neighbour sitting side by side on chromosome 1, happened much later, in a primate ancestor, in the deep history of the great apes, long before there was anything you'd call a human. By the time our own species walked the earth, this whole arrangement was already old, already settled, already humming away in the red cells of every one of our forebears. So here is the correction, and it's a sharp one. It is simply wrong to frame Rhd-negative blood as something uniquely strange to humans, as if every other creature flies the flag and only some odd subset of people lost it. The flag, and the machinery behind it, are universal and ancient, shared with fish and worms and bacteria, conserved across an almost unimaginable depth of time. What is genuinely peculiar is not the protein, it's the deletion, the specific human event in which the gene for the D-flag was cut out and then, against all the rules, allowed to spread. That is the real anomaly. Not an ancient protein, those are everywhere, a relatively recent loss of one in our species that should have been fatal to itself and wasn't, which means the wild stories have it exactly upside down. The mystery is not that Rhd-negative people carry something strange and other, it's that they carry the absence of something almost universal, and that the absence didn't die out. Keep that the right way up in your mind and you'll never be fooled by the alien version again. The strangeness isn't in the people, it's in the survival of a gap. So when and in whom did the gap first open? Let's go and find the source. This is the part of the story where almost everything you've heard online lives, and where this telling is going to walk a very deliberate path. We're going to take the famous theories one at a time, in good faith, and ask of each one a single fair question. What does the actual evidence support? Not what feels exciting, not what we'd like to be true, what holds up when you touch it. And the spirit of this matters. The point is not to mock anyone who has found these ideas compelling. The pull of them is real and human, and we'll talk later about exactly why. The point is that you deserve honesty, and the honest answer, in this case, turns out to be more remarkable than the inventions. So let's clear the ground. Start with the most respectable of the theories, the one that even sounds scientific. The Neanderthal idea. It goes like this. Modern humans, after leaving Africa, met and interbred with Neanderthals in Europe and the Near East. We know this is true, it's one of the great confirmed discoveries of the last couple of decades. Most people whose ancestry runs through Europe and Asia carry a small percentage of Neanderthal DNA, woven into their genome from those ancient encounters. So the theory goes that maybe Rhd-negative blood is part of that inheritance. Maybe the deleted gene is a Neanderthal gift, a trace of that older, vanished kind of human, surfacing in 1 in 7 of us. It's a beautiful idea. It connects the strangeness of the blood to the strangeness of our deep family tree. It's also, almost certainly wrong, and the reason it's wrong is a small masterpiece of detective work. Remember what we found on the map, that Rhd-negative blood occurs in Africa too, and that the most common European deletion, the clean cutting out of the gene, is also found among Africans. Now follow the timeline. The interbreeding with Neanderthals happened after modern humans had already left Africa, because that's where Neanderthals were, out in Europe and Asia, not in Africa. So if the deletion came from Neanderthals, it should be a European and Asian thing, picked up on the way out. It should not be sitting in African populations who never met a Neanderthal, but it is. According to the geneticists who specialise in this very system, including one of the top authorities on Rh genetics at the United States National Institutes of Health, the most likely story is that the deletion arose in Africa before our ancestors ever left, and was simply carried out with them when they migrated. The deletion is older than the Neanderthal encounters. The timing rules it out. You cannot inherit from Neanderthals something you already had before you met them. There's a famous version of the claim that pins it to a specific number, that Rhd negative blood came from Neanderthals about 35,000 years ago, and the verdict from the people who actually study this is blunt. That claim is very likely incorrect and far too recent. The gap in the gene is older and deeper than that, and it traces back not to a different kind of human, but to our own shared African origin. If Rhd negative blood points to any kind of common ancestor, it points backward, toward the single source from which all of us came, not sideways toward some separate or exotic lineage. It is, if anything, a mark of our deep unity, not our division. Then comes the bigger, wilder family of theories, the ones that get the most views and the least evidence. That Rhd negative blood is the trace of extraterrestrial visitors who interbred with early humans. That it marks descent from the Nephilim, the mysterious giants or sons of God, named in a few lines of ancient scripture. That it belongs to the survivors of a drowned continent, Atlantis or Lemuria, or whichever lost world a given storyteller prefers. Let's be straight with each other the way I'd want someone to be straight with me. There is no evidence for any of this. None. There's no genetic signature of a separate non-human source mixed into Rhd negative people, and we have now read these genomes in extraordinary detail. If a fingerprint like that were there, we would see it, and it isn't. There's no drowned city, and no sunken continent, and not a single piece of archaeology pointing to the lost homeland these stories require. There is no extraterrestrial anything. These ideas are built on top of the real puzzle the way ivy grows up a real wall. They cling to something solid, they use its shape, and from a distance they can look like part of the structure. But they are not part of it. Pull them away and the wall is unchanged. The genuine mystery, why a costly gene survived, was never explained by any of them. They just attached themselves to it because it was there, and unexplained things attract stories the way still water attracts insects. I mention these theories at all, rather than ignoring them, for one reason. They are everywhere, and you have almost certainly encountered them, and treating you as someone who can handle the plain truth is more respectful than pretending the question is open when it isn't. The blood is genuinely mysterious. The aliens are not part of the mystery. Both of those things are true at once. And then there's the most seductive of the homegrown myths, the one wrapped in velvet and crowns, the idea of blue blood. The notion that Rhd negative blood is the blood of royalty, that the old aristocratic families of Europe carried it at unusual rates, that it marks a superior, set-apart lineage, that blue blood was literally about something special in the veins. There's a thread of fact here, and it's worth untangling carefully, because the truth turns the myth completely inside out. It is true that some European royal and noble lines show elevated rates of Rhd negative blood and of various other uncommon traits, but the reason is the exact opposite of special. The reason is inbreeding. For centuries, European royalty married within a tiny pool, cousins marrying cousins, uncles marrying nieces, the same few families circling each other generation after generation to keep crowns and territories in the bloodline. And inbreeding does something very specific to genetics. It concentrates whatever is already in the family. Not the good traits selectively, not the noble traits, but whatever is there, good and bad alike, doubled up and pushed to the surface because there's no fresh genetic material coming in to dilute it. So if a royal line happened to carry the Rhd negative deletion, intense inbreeding would indeed concentrate it. But that's arithmetic, not destiny. It's a mechanical consequence of marrying your relatives, not a sign of superior blood. And we know exactly how that arithmetic looks when it goes wrong, because the same royal families are the textbook cautionary tales. The Spanish branch of the Habsburg dynasty inbred so relentlessly that the traits they concentrated were catastrophic. Most infamously a jaw so pronounced and deformed generation after generation that the last Habsburg king of Spain could barely chew his food, and the line finally collapsed into infertility and early death. That's the jaw of the Habsburgs. And it's not a mark of glory. It's a warning carved into a royal face. Or take Queen Victoria, from whom a single mutation for haemophilia, a disease that prevents blood from clotting, spread through the intermarried royal houses of Europe into the thrones of Spain, of Germany, of Russia. It rode in the veins of the Russian heir whose suffering helped pull a mystic named Rasputin into the imperial court in the last years before that whole world came crashing down in revolution. So when someone tells you Rhd negative blood is the blood of kings, the honest reply is that in the cases where royals concentrated it, They did so by the very same process that gave them deformed jaws and bleeding heirs. It is not the signature of a superior bloodline. It is, if anything, a multi-generational warning about what happens when a population stops mixing and starts folding in on itself. The opposite of magic, just consequences. So strip all of it away, the Neanderthals, the aliens, the Nephilim, the sunken continents, the crowns, pull off all the ivy. What's left standing on the wall? Something quieter, and to my mind far stranger and more beautiful than any of the inventions. A single genetic deletion, a short stretch of DNA simply gone from chromosome 1, the kind of slip the genome's own architecture made almost inevitable, and everything we can currently read in the genes points the same way. It most likely arose somewhere in Africa, in deep prehistory, in some person we will never know, long before our ancestors fanned out across the world. It rode out of Africa in the blood of those who left, and rode on too in the blood of those who stayed. It travelled in human bodies through every migration, every catastrophe, every generation, for tens of thousands of years. And in certain sheltered corners of the world it didn't merely survive. It climbed, against the gravity of natural selection, to frequencies it had no business reaching, peaking in those Atlantic mountains where the language stands without a single living relative and the blood runs higher than anywhere else alive. No spaceship, no drowned kingdom waiting to be remembered, and nothing hidden in the blood of kings. Just a missing piece of code, and a question it has been asking us, in silence, for 90 years. And the question is, the one we've circled the whole night, and can no longer put off. Why didn't it die? We've cleared the ground. Now we have to face the cost directly, and feel its full weight, before we can understand why anyone thinks it survived. To understand why scientists call this an enigma, why it isn't just a curiosity but a genuine, stubborn problem that won't resolve, you have to feel what this trait actually cost for thousands of years, before anyone on Earth understood what was happening. Numbers won't do it. We have to sit for a little while with the human shape of it. So, a woman, in any century you like before the very recent past. Pick a quiet one, a farming village, a stone house, the ordinary rhythm of a life lived close to the ground. She is Rhd negative, though of course no one in her world has any concept of such a thing, any more than they have a concept of a galaxy. She marries, she becomes pregnant, and her first child is born healthy, pink, squalling, whole. The relief and joy of it. She nurses the baby, she watches it grow, she has every reason in the world to expect that the next one will be the same. What no one can see is that during that first birth, in the mingling of blood that birth involves, her body met the baby's D-flag for the first time, that one inherited marking the baby carried and she did not, and her immune system, doing exactly what it was built to do, began, quietly, to learn it as an enemy. By the time she's pregnant again, she's no longer the woman she was. Her body is armed now, carrying a memory she never chose and cannot feel. And so the second child is born wrong, yellow with jaundice, listless, struggling. Or born too soon, already dying. Or it never reaches birth at all, and there is no explanation to be had. The midwife has none, the priest has none, and neither does the wise woman or the physician or anyone else she can reach. Just the unbearable arithmetic of it. The first one lived, the second one did not, and no one can tell her why. And then, often, the third goes the same way, and the fourth? A particular family becomes known, in the quiet, cruel way villages know these things, as one where the babies don't live. And the cause, the whole time, is invisible. A single missing flag and the long, faithful memory of an immune system doing its job too well. Read the old parish records, the registers of births and burials kept in churches across Europe for centuries, and you can find this grief without knowing its name. Babies baptized and buried within days. The same surname recurring in the burial column year after year. People reached for the explanations they had, God's will, a punishment, an imbalance of humours, the evil eye, something the mother ate or did or failed to do. Whole frameworks of meaning, of blame and penance and dread, were built in part over a gap that no one could see, on the short arm of a chromosome no one knew existed. The suffering was real, and the explanations were all wrong, and there was nothing to be done. And here is the thing that turns private grief into a scientific problem. In the populations where Rhd negative blood was common, those Atlantic peaks, that 1 in 3 among the Basque, This was not a rare misfortune striking the occasional unlucky family. It was woven into the fabric of reproduction itself. In every generation across those communities, it touched a real and substantial fraction of pregnancies. The cost wasn't a freak event at the edge of the population. It was a steady, grinding tax on having children, levied generation after generation, exactly where the trait was most common. Hold that against the cold logic of evolution one more time, because this is the hinge of the entire story, and now you can feel it instead of just thinking it. A trait that does this should bleed out of the gene pool. Every later child lost is a copy of the gene removed from the future, and worse, removed precisely from the families that carried the most of it, where two RHD negative prone lines met. The downward pressure isn't gentle, it's severe, and it's aimed with terrible precision at the one thing evolution cares about above all else: surviving offspring. Over enough generations, a trait this costly should collapse towards zero. It should become a rare genetic disease, like so many others, a thing that crops up occasionally and is constantly pruned back. It should be a footnote. Instead, it became a third of the Basque, a quarter of the Irish, 15% of all of humanity. It rose against the gravity, and it didn't just cling on in one place by luck. It reached high frequencies across whole regions, stably, for thousands of years, refusing both to vanish and to take over completely. That stubborn, stable persistence in the teeth of a real reproductive cost is the entire puzzle. It is what makes geneticists, sober people, who don't reach for the word lightly, call this an evolutionary enigma. Something was holding the trait up against a force that should have driven it down. The whole 100-year search has been to find out what. But before we go looking for the answer, before we reach the real frontier, there is one more thing to tell about the cost. Because the story of the cost has an ending, not an explanation. We'll see that the explanation never came, but a rescue. After all those thousands of years of invisible, unanswerable grief in the span of a single decade in the 20th century, human beings figured out how to stop it. And that part of the story deserves to be told in full, because it is one of the quiet, total victories of medicine, and because it is recent enough to touch. By the middle of the 20th century, the mechanism we walked through earlier was understood. Doctors knew what was happening. The RHD-negative mother, the RHD-positive baby, the immune system learning the D flag at the first birth and arming itself against the next. They could even, with effort, predict which pregnancies were at risk. But knowing why a thing happens and being able to stop it are two very different powers, and for a long time, medicine had only the first. Doctors could watch the danger coming and do almost nothing to prevent it. They could deliver early, attempt crude transfusions, manage the aftermath, but the fundamental event, the mother's body learning to attack, they could not head off. They were still mostly spectators to an old grief. The breakthrough, when it came, was beautiful in its simplicity, almost a trick, a piece of immunological judo that turns the problem against itself. And it came, as the best discoveries often do, from several directions at once, from people who were rivals as much as collaborators. In New York, at Columbia, an obstetrician named Vincent Frieda and a blood bank director named John Gorman were chasing it. Working with them was a research scientist named William Pollock at a pharmaceutical lab. Across the Atlantic in Liverpool, two physicians named Cyril Clarke and Ronald Finn were pursuing the very same goal, independently, racing toward the same idea from the other side of the ocean. In Canada, others, among them a paediatrician named Alvin Zipursky, and later Bruce Chown and John Bowman, were laying down crucial pieces, working out just how much of a baby's blood actually crosses into the mother and when. It was, in the way of real science, less a single eureka than a convergence. Several careful minds in several cities circling the same insight until it came clear. And the insight was this: the whole tragedy depends on one step, the mother's immune system learning the D flag and building its own lasting memory of it. If you could prevent that one step, if you could stop her body from ever learning, then there would be no armed antibodies waiting, and the next baby would be safe. So how do you stop an immune system from learning something? Here's the elegant part. You give it the answer before it can work out the question. Right after the birth, before the mother's own immune system has had time to notice and respond to the baby's stray red cells, you inject her with ready-made antibodies against the D flag, antibodies made by someone else, borrowed, handed to her fully formed. Those borrowed antibodies sweep through and quietly clear away the baby's leftover blood cells, the ones carrying the foreign flag, before her own immune system ever gets a proper look at them. The intruder is gone before the alarm can sound, and because the threat is removed so fast, her body never builds its own response. It forms no lasting memory, and it makes none of the antibodies that would endanger the next child. One injection, the whole chain broken at its first link, the next pregnancy and the one after that made safe. They called it Rh immune globulin. The brand name that became famous was Rogam, and in 1968, within living memory, more recent than the moon landing was distant from it, it was approved and went into use. There's a person at the centre of that moment, and her name deserves to be said because she stood right on the line between the old world and the new. Her name was Marianne Cummins, from New Jersey. In 1968, she was 29 years old and pregnant with her third Rhd-positive child, exactly the situation that, in an earlier era, carried such danger. She had read in a newspaper about a new treatment that was about to be released. There was a problem. The drug's release date was just after her due date. If she delivered on time, she would miss it by a single day. As it happened, the timing worked, and she became the first person to receive the approved treatment, hours after it became available. She went on to deliver a healthy baby. One woman standing at the exact hinge of history who got there with a day to spare. The effect of the treatment, where it became standard, was not gradual. It was a collapse. In the United States alone, hemolytic disease of the newborn from this cause had been killing on the order of 10,000 babies a year, and damaging many more, leaving survivors with brain injury and lifelong disability. Within about a decade of Rogam becoming routine care, deaths from the disease fell by more than 90%, a condition that had haunted our species for longer than we have had writing, that had filled those parish burial registers, that had broken those families in the stone houses, was, in the developed world, very nearly switched off. The people who developed it were eventually honoured with one of medicine's great prizes, and a major magazine named it among the top medical achievements of its decade. But the real monument is the simple, staggering arithmetic. Countless children alive who, a few years earlier, would not have been. I want to pause here, on this line through time, because it is close enough to touch, and because if you are Rhd negative, or if you love someone who is, this is the part of the story where you might find your own family standing. There are women alive today who are among the last to bear children before the injection existed. Women who lost a first baby, or a second, or a third, and were told, the way mothers had been told for 10,000 years, that there was nothing to be done, and who then, perhaps, lived just long enough to see the world change and to have a later child survive who, a few years earlier, would not have. There are far more women, likely most of the mothers you know, who are Rhd negative, who came just after the line. They got a single injection during a routine pregnancy, barely thought about it, maybe don't even remember it, and carried 2, 3, 4 healthy children into the world, never knowing how close they stood to the older, crueler version of the same story. The same blood, the same bodies, separated by a few short years and one quiet discovery. If you are here in the world because of that injection, if your own existence, or your children's, runs through that needle and those borrowed antibodies, then you're standing on the near side of one of the sharpest, kindest lines that medicine ever drew. It's worth a moment of something like awe in the dark for the ordinary miracle of having been born on the right side of it. There's a shadow on this triumph, though, and honesty asks us to name it. That line through time isn't only a line between then and now, it's also a line across the map, and not everyone lives on the lucky side of it. The collapse in deaths happened where the treatment became routine, in wealthy countries with reliable hospitals, with the injection in stock and given on schedule. In much of the world, that's still not guaranteed. The same condition that has been all but switched off in the developed world remains, in poorer regions with thinner medical systems, a real and ongoing cause of stillbirth, of newborn death, of brain damage in the babies who survive it. The biology is identical everywhere. The missing flag, the armed mother, the endangered later child. What differs is only whether a particular small vial of borrowed antibodies happens to reach a particular mother in time. It's a sobering reminder that the rescue we just celebrated is, for now, a privilege of geography and circumstance as much as a gift of science, and that the old, crueler version of this story has not ended for everyone. It has only ended for some of us. That too is part of the truth of this blood. But here is the thing that matters most for the mystery we're following, and it's a quiet, sobering point. Notice what medicine did, and notice what it did not do. It removed the cost. In the present, in the places where the treatment reaches, the danger is gone. That is an enormous good. But removing a cost in the present is not the same as explaining how a trait survived that cost in the past. The injection arrived in 1968. The trait is tens of thousands of years old. For all those millennia before, the cost was fully in force, real and severe, and aimed at reproduction, and the trait survived anyway, rose anyway, reached a third of a population anyway. Medicine answered the practical question, the merciful one. How do we stop the harm now? It did not answer, and could not answer, the deeper one that has nothing to do with us or our hospitals. How did this blood survive the harm for all the long ages when no one could stop it? That question was waiting, untouched, the whole time, and it is to that question, the real frontier, the genuine open edge, that we finally turn. So here we are, at the edge. Not at a manufactured mystery dressed up to look deep, but at the genuine frontier of what is known, where the people who study this for a living will tell you, in plain words, that they have ideas and not answers. This is the place the whole night has been walking toward. Let's arrive slowly. For about 90 years since the trait was first discovered, in that laboratory full of monkey blood, the persistence of Rhd negative blood has been called, in the scientific literature, an evolutionary enigma and an evolutionary riddle. Not by mystics or storytellers, by geneticists. The word enigma is theirs. It turns up, along with riddle and puzzle, in the serious scientific literature on this very question. These are people whose whole training is to drain mystery out of things, to replace wonder with mechanism, and even they, looking at this, reach for the language of riddles. That should tell you something. When the skeptics call a thing a mystery, the mystery is real. Let's be precise about why it's a riddle, because the precise version is more interesting than the vague one. The problem isn't just that a costly trait survived, it's the particular way it survived. Both versions of the gene, the working version that makes the D flag and the deleted version that doesn't, coexist side by side in the same populations stably for thousands upon thousands of years. Neither one wins, neither one disappears. And in biology, that specific situation, two versions of a gene refusing to eliminate each other over long stretches of time, is itself a clue. Because normally, one version should win. If the negative version is costly, the positive version should slowly push it out. If for some reason the negative version were favoured, it should slowly push the positive out. A stable standoff, lasting millennia, usually means something is actively protecting both. Something is rewarding having a mix. Geneticists have a name for this, balancing selection. And the entire search has been to find the thing doing the balancing, the hidden hand keeping both versions in play when the simple math says one should have won. So researchers went looking. Let me walk you through the search, because the search itself is the real story here, not a tidy answer, but the honest shape of people hunting for one. The first and most obvious idea was that maybe Rhd negative blood quietly protects against some disease. This is not a wild guess, it's exactly how other strange blood traits work. The most famous example is sickle cell, a trait that, in its double dose, causes a serious disease, but in its single dose protects powerfully against malaria. In regions where malaria is a killer, that protection is worth so much that the trait persists despite its cost. Held up by the disease it defends against, it's a perfect proven case of a harmful-looking gene being maintained because it secretly helps. So the natural hypothesis was that maybe the Rhd deletion does something like that. Maybe it shields its carriers from some illness, and that hidden shield was worth more across history than the cost in lost pregnancies. In 2012, a team set out to test this directly. They took genomic data from several human populations and went looking for the specific fingerprint that this kind of protective benefit leaves in the genome, the telltale signature of an allele being actively favoured, swept upward by natural selection because it helps. If Rhd negative blood were the new sickle cell, protecting against some disease X, that signature should be there to find. They didn't find it. No clear signal of that classic kind of positive selection on the deletion, which doesn't completely close the door since absence of a clear signal isn't quite the same as proof of absence. But it means that the simple, satisfying story, the Rhd negative is secretly good against some plague story, has not shown itself in the data. If there's a straightforward hidden benefit of that kind, it has stayed well hidden. The obvious answer came up empty, and that empty result is what pushed researchers toward the second idea, the strongest lead anyone currently has, and one of the strangest things in all of human biology. It involves a parasite. And it shifts the whole question in a way that takes a moment to absorb. Here's the shift. What if the thing being protected isn't being negative at all, and isn't being positive either? What if the advantage belongs to the people who carry one of each? Remember the recessive genetics from earlier, that to actually be RhD negative you need 2 deleted copies, while plenty of people walk around with 1 deleted copy and 1 working copy, flying the D flag normally, never knowing they're carrying the deletion at all. Those people are the heterozygotes, 1 of each. And the leading hypothesis for this whole 100-year riddle is that, in a particular and surprising way, the heterozygotes are the winners, and that their advantage is what keeps both versions of the gene perpetually in circulation, neither ever allowed to disappear. The researcher most associated with this idea is a Czech biologist named Jaroslav Flieger, who has spent much of his career on a creature you may have heard of in another context, Toxoplasma gondii. It's a single-celled parasite, and it is astonishingly common. Depending on the country, somewhere between roughly 1/5 and 1/2 of all people carry it, usually for life, usually without ever knowing, tucked away quietly in the tissues, including the brain. You can pick it up from undercooked meat, or from contact with cat feces, since cats are the parasite's preferred home. For most people, a latent toxoplasma infection causes no obvious illness at all. But no obvious illness is not the same as no effect, because this is the unsettling thing about latent toxoplasma. It appears to subtly change its hosts, quietly, at the edges. One of the most consistent findings is that people carrying it have measurably slower reaction times, a little slower to respond, a fraction of a second behind, in ways you'd never notice in daily life, but that show up clearly on a test, and that may show up tragically in things like a slightly raised rate of traffic accidents. The parasite seems to dull the reflexes just a little, in a way that accumulates across a population of millions into something real. Now here is Flieger's finding, the one that could crack the riddle, and it carries a twist we have to get exactly right, because it is very easy to get backwards. He and his colleagues looked at how this toxoplasma-induced slowing interacted with people's RhD genetics, and what they reported was this. Among the people carrying the parasite, the ones who held steady, whose reaction times barely suffered despite the infection, were the heterozygotes, the 1 of each people, 1 working copy of the gene, 1 deleted copy. And here is the part to hold carefully in the dark. A heterozygote, carrying even a single working copy, flies the D flag, which means the protected ones are not the RhD negative people at all. They are a particular kind of RhD positive person, the ones who test positive, who carry the flag without a thought, and who quietly hold 1 deleted copy hidden underneath it. And the RhD negative people, the ones with 2 deleted copies, the very people this whole story keeps circling back to, the ones who might be lying in the dark right now, were, if anything, on the vulnerable side of it. There's a strange, almost rueful detail buried in the numbers here, and it's worth giving to you straight because it's truer and kinder than any flattery. When there's no parasite involved at all, RhD negative people seem to have the quickest reaction times of anyone. They start out ahead. But under infection, that lead collapses. They slow the most, and in one large study of military drivers, infected RhD negative men ran something like 3 times the risk of a traffic accident. The edge they're born with is real, and the parasite is very good at taking it away. The heterozygotes, meanwhile, those hidden carriers, flying the flag with a single deletion tucked behind it, kept their footing whether the parasite was there or not. And so the deletion survives, on this account, not in the people who visibly carry it, but in the far larger invisible crowd of positive testing carriers who never know they hold it, the ones in whom being a little of both turns out to be the safest thing to be, sit with what that would mean if it holds. In a world saturated with this parasite, and our world is saturated with it, and was even more so in the past, when people lived closer to cats and ate rougher food, there's a quiet, ongoing advantage to carrying both versions of the gene. The heterozygotes, the 1 of each people, would be a little sharper, a little safer, a little better protected against an infection most of them are carrying without knowing it. And if the heterozygotes are the favoured ones, then the population has a powerful reason to keep both versions of the gene around. because you can only make heterozygotes if both versions exist. The working copy can't disappear because you need it to make the protected mix. The deleted copy can't disappear for the same reason. Each one is preserved not for its own sake, but because it's half of the winning combination, and that would explain the thing nothing else could. Why neither version ever wins. Why the trait that should have bled to zero didn't, and the trait that might have taken over didn't either. Why this strange stable standoff has held for thousands of years. The cost in lost pregnancies is real, but it might be offset, balanced by the hidden advantage of being the mix, in a world full of a mind-altering parasite. The blood that should have disappeared survives, on this account, not despite the standoff but because of it. The very fact that there are two versions is what keeps both alive. There's even a tantalizing further wrinkle, which is that this could help explain the map, the geography we found so strange. Toxoplasma isn't evenly distributed across the world. Some regions have far more of it than others, and if the advantage of the gene mix depends on the local burden of the parasite, then the frequency of Rhd-negative blood might track in part with the local history of toxoplasma. Some studies comparing many countries at once have reported patterns broadly consistent with that idea, disease burden lining up with blood type frequencies in the directions the balancing selection story would predict. The pieces, when you squint, seem to want to fit together. A parasite, a gene mix, a buffer against its effects, and a map shaped by where the parasite was worst. But now I have to do the thing that this whole telling has been built to do, the thing that separates an honest story from a seductive one. I have to tell you plainly that this is not settled. It is the leading hypothesis. It is not a proven fact. The studies behind it are real, published in real journals, and they are also debated. Some of the effects have been hard to replicate cleanly. When researchers, including Fleger's own group, have gone back to test the idea again, more rigorously, with larger samples and pre-registered methods designed to keep anyone from fooling themselves, the picture has come back mixed. Some support, some complications, nothing that closes the case. The effect sizes are subtle. The biology connecting a missing red cell flag to a parasite in the brain is not fully worked out. We don't have a clean mechanism that says here is exactly why one of each protects you, and on top of all that, the parasite itself is in retreat. In many developed countries, the rate of toxoplasma infection has been falling steadily, as people eat more carefully and live further from the old exposures, which means that even if this advantage was once a powerful force, it may be quietly switching off in our own time, fading along with the parasite that drove it. The hidden hand may be lifting from the scale just as we've started to notice it was there. So the honest state of the science, here in our own moment, is this. We have a genuinely promising lead, strange and elegant and partly supported, and we do not have a final answer. After 90 years, with all our genomes sequenced and all our statistics sharpened. The riddle is narrower than it was, since we can say what kind of answer it probably is, a balancing act rather than a simple benefit, but it is not closed. Someone may close it in your lifetime. It might be closed by the time some of you listen to this again. But tonight, as you lie there, it is open. The question is still out there in the dark, unanswered. The same as it was for Landsteiner, the same as it will be for the children of the people now studying it. And there's one last open piece to fold in, because it makes the whole thing complete in its strangeness. We talked near the start about how we don't even fully know what the D protein is for, that it's some kind of gas channel, moving ammonia or carbon dioxide, its exact role still not pinned down. Put that together with everything else, and look at what sits at the centre of this story. A protein whose precise purpose we cannot state, missing entirely from 1 in 7 of us, by way of a deletion the genome was almost built to make, carried out of Africa in deep prehistory, surviving a reproductive cost that should have erased it. Held up, we suspect but cannot prove, by a brain parasite and the quiet advantage of being a genetic mix. Mapped to a peak in a mountain, people whose language has no living relatives anywhere on Earth. That's the real mystery. Every piece of it is true. It needs no alien, no drowned continent, not a single drop of royal magic. The honest version, told straight, with all its gaps left open, is stranger and richer than anything the myths ever managed to invent. And that's worth remembering when we turn, now, to the myths themselves, and to the very human reasons they hold on so hard. Before we leave that frontier behind, it's worth lingering for a moment on the strangest character in the whole story, the parasite that might, just might, be the hidden hand on the scale. Because once you understand what Toxoplasma gondii actually is, The idea that it could be shaping the blood of 15% of humanity stops sounding far-fetched and starts sounding almost inevitable. This is a creature that manipulates minds for a living. Here is how it makes its way through the world. Toxoplasma can live in many animals, but it can only complete its life cycle, only truly reproduce, inside a cat. The cat is its home, its destination, the one place it needs to get to. So the parasite has a problem. If it finds itself inside, say, a rat or a mouse, it needs that rodent to be eaten by a cat so it can reach the only place it can finish its life. And evolution's solution to that problem is one of the eeriest things in all of biology. The parasite lodged in the rodent's brain appears to change the rodent's behaviour, and not randomly. Infected rodents lose their normal, hardwired, life-saving terror of cats. Some studies suggest they become almost drawn to the smell of cat urine, the very scent that should send them fleeing. The parasite in effect rewrites the survival instincts of its host to deliver that host into the jaws of the cat it needs to reach. A single-celled organism, with no brain of its own, reaching up into the brain of a mammal and quietly turning its deepest fear into attraction. It is hard to read about and not feel a small chill, and this same parasite is carried latently by somewhere between 1/5 and 1/2 of all human beings, depending on where you live. We are not its intended destination. We're a dead end for it, an accident, a host it can't actually use to reach a cat. But it gets into us all the same, from undercooked meat or from the soil and surfaces a cat has touched, and it settles in, including in the human brain, usually for life, usually without our ever knowing. And the unsettling thread of research we touched on earlier, the slowed reaction times, the subtle shifts, is the question of whether this mind-altering passenger, so good at nudging the behaviour of rodents, is also, gently, nudging ours. Most scientists are cautious here, and rightly so. The human effects, where they appear, are subtle and contested. But this is the creature at the centre of the leading explanation for our riddle. Not a tidy little microbe. A practised manipulator of nervous systems, riding in 1/3 of humanity, possibly tuning the very reflexes that the RHD gene mix may help protect. Put it that way, a brain parasite and a blood gene, locked in some ancient half-glimpsed balance across thousands of years, and the truth really is stranger than the aliens. It's just stranger in a way you can actually check. Let me turn the whole thing, for a moment, away from the species and toward you. Because the question that's been quietly running under everything tonight, whether this might be you, has a real answer, written in the simple genetics of families, and it's worth understanding plainly. You carry 2 copies of the RHD gene's location, one from your mother, one from your father. To actually be RHD negative, to show the trait, you need the deleted version from both. Which leads to something many people find surprising, even unsettling when it shows up in their own family. 2 RHD positive parents can have an RHD negative child. It happens all the time, and it's not a sign of anything amiss. It simply means both parents were quietly carrying one deleted copy along with their working one, flying the D flag themselves, never knowing, until 2 of those hidden copies happen to meet in a child. Whole families discover the trait this way, arriving as if from nowhere in a single child, when in truth it had been travelling silently through the line for generations, carried by people who showed no sign of it at all. That child isn't a stranger in the family. That child is the place where a long-hidden thread finally became visible. And it means the question of whether you carry this isn't quite as simple as positive or negative on a form. Even if you fly the D flag perfectly well, you might be one of the carriers, one of the heterozygotes, holding one working copy and one deleted one, part of the very genetic mix that, if the parasite story holds, may be the quiet reason the whole thing survives. You could be a link in the chain without ever appearing on the map. The trait could be in your blood, or in your children's future, or sleeping in your family tree waiting for the next time 2 hidden copies meet. Most people never find out. They go their whole lives, like nearly everyone through all of history, never knowing which version they carry or what it might mean. There's something fitting in that a mystery this old should still, for most of us, begin at home, unanswered, in our own veins. Which brings us finally to the stories and to why. Faced with all this genuine strangeness, so many people reach instead for ones that aren't true. Of all the pieces of human biology, and there are thousands you could pick, why does this one attract more myth than almost any other? Why is it RHD negative blood, specifically, that has spawned the forums and the late-night videos and the lists of secret traits, when nobody builds a mythology around, say, their Kell antigen or their Duffy type. That question has an answer, and the answer turns out to be about us, about how human minds work, far more than it's about the blood. So let's look honestly at the stories, take them seriously enough to actually weigh them, and then understand why they stick. Start with the famous list of Rhd negative traits, the supposed superpowers. If you go looking, you'll find them recited again and again with small variations. That Rhd negative people have higher IQs, that they have piercing, often light-coloured eyes, that they run a lower body temperature or lower blood pressure, that they're unusually sensitive to light or to heat or to electromagnetic fields, that they have heightened intuition, vivid dreams, a feeling of not quite belonging, and, at the far end, outright psychic ability. It's quite a list. Held up to the light of careful evidence, almost all of it dissolves, but it dissolves in interesting ways, so let's go through it rather than just waving it off. Take the IQ claim, probably the most repeated of all. When you do the thing that separates real findings from noise, large studies, careful controls, accounting for the fact that the populations where Rhd negative blood is common also tend to differ in education, wealth, and 100 other things that affect test scores, the supposed intelligence gap simply melts away into statistical static. There's no robust, replicated finding that the missing D flag makes anyone smarter. What there is the shared ancestry effect we'll come back to in a moment, plus the universal human tendency to remember the flattering claim and forget the boring correction. The eye colour and pale skin traits are a perfect clarifying example, because here there's a real correlation hiding a false cause. It is true that, on average, Rhd negative people are somewhat more likely to have light eyes and pale skin, but the missing D flag doesn't cause light eyes any more than light eyes cause the missing flag. They simply travel together, because they're common in the same populations, the peoples of Europe, especially that Atlantic fringe, where light eyes, pale skin, and the Rhd deletion all happen to be frequent. They're fellow passengers, riding in the same ancestral populations, not cause and effect. It's like noticing that people who speak Norwegian are more likely to enjoy cross-country skiing and concluding that the language teaches the sport. Shared origin, not hidden power. Pull the trait out of its population, and the superpower goes with it, because it was never in the blood to begin with. It was in the family tree. The lower blood pressure and lower temperature claims are the kind where you have to be fair in both directions. A handful of small studies have hinted at tiny average differences in this or that physiological measure. But a tiny average difference that shows up in some small studies is a universe away from a different kind of organism, which is how the myth tends to phrase it. The body of an Rhd negative person is, in every way that matters, an ordinary human body. There is no second species walking among us. There's a missing flag on one kind of cell, and psychic ability, the claim at the far romantic end, has never survived a single controlled test. Not one. What has been demonstrated repeatedly and robustly is something quite different and quite human. People who already believe they're special are very good at noticing the coincidences that seem to confirm it, and very good at forgetting the ones that don't. A vivid dream that came true, a moment of intuition that landed. We all have those. We just don't all build an identity around them. The psychic finding isn't a finding about blood. It's a finding about belief. There's one health claim in this family that deserves much more careful treatment than the rest, because it's surged enormously in recent years and because, unlike the psychic stuff, it has a real grain of something at its centre. It's the claim you'll hear constantly. I'm Rhd negative and I never get sick. I never even caught COVID. This one we have to handle honestly, because there's genuine science here, wrapped in a lot of wishful thinking, and the truth is right in the difficult middle. Here's the careful version. During the pandemic, several large studies did look at whether blood type related to COVID risk, and a few of them did find a small signal around Rh status. A study in the New York hospital system reported that Rh negative people seem to have a slightly lower risk of getting infected, of needing a ventilator, of dying. A very large Canadian population study, looking at well over 200,000 people, similarly found that O and Rhd negative blood were associated with a slightly lower risk of infection and of severe illness. So it's not nothing. There may be a small tilt in the odds, but, and this is the essential but, other large studies, including ones from elsewhere, found no real effect once you carefully accounted for age, health, and other factors, or found that the effect vanished when you looked only at people who were already infected. The findings, taken together, are genuinely mixed. Rh status has been studied less than the ABO types. The effects where they appear are small, and they don't line up cleanly across all the data. So the honest summary is maybe a slight statistical nudge in some populations, maybe nothing once you control properly, and the studies disagree with each other. What it absolutely is not is immunity, and this is where the gentle but firm correction matters, because lives can ride on it. No blood type makes anyone immune to COVID or to any other infection. RhD negative people caught COVID. They got seriously ill. Some of them died. A small statistical difference spread across a population of millions is not a shield around any single person. It cannot be. That's not how population statistics work. If you personally went through the whole pandemic untouched, that is genuinely wonderful, and you're allowed to be glad of it. But it wasn't your blood type standing guard at the door and treating it as armour, as a reason to skip the ordinary proven things that actually protect you and the people around you, would be to mistake a faint background tilt for a personal force field. The blood is interesting. It is not armour. Both of those can be true, and holding them together is exactly the kind of honesty this whole story is built on. Then there's the one the comment sections love most, and this one is real and worth the wonder. The so-called golden blood. Because beyond the simple split of positive and negative, beyond the single missing D flag, there exists something at the absolute far edge of rarity. Remember that the ARC system isn't just the one flag. It's a whole family, more than 50 distinct antigens riding on those proteins. Almost everyone has most of them. But there are a tiny number of people on Earth who are missing all of them. Not just the D flag. Every Rh flag, the entire set, simply absent. This condition is called ARD null, and it is so rare that it has earned the nickname golden blood. How rare? It was first identified in 1961 in an Aboriginal Australian woman, and in all the decades since, fewer than 50 people in the entire history of the world have ever been documented with it. At any given time, the number of active donors, original people willing and able to give blood, can be counted on your fingers, often fewer than 10 on the whole planet. Before 1961, in fact, doctors assumed that a person missing all those antigens simply couldn't survive to be born at all. And the reason it's called golden is genuinely poetic and genuinely double-edged. Because Renal blood lacks all those flags, it can be given safely to almost anyone with a rare Rh blood type. It's universally compatible within the Rh system in a way no other blood is, which makes it precious beyond price to certain rare patients in crisis. A person with golden blood can be a lifesaver for strangers across the world, but the gift runs only one way. Because their blood has none of those flags, and a renal person's immune system has never learned any of them, which means they can safely receive blood only from other renal donors, from those few people. So for the person who has it, golden blood is not a superpower at all. It's a kind of beautiful isolation. Many of them bank their own blood, freezing it away against the day they might need it. Because if an emergency struck and they needed a transfusion, the supply of compatible blood in the entire world might be a handful of frozen units and a phone tree of fewer than 10 people. It is rare. It is remarkable. And for the one carrying it, it's a genuine medical burden, the opposite of a gift. And like so much here, it's far rarer than the number of people online who are quite sure they have it. So why? Why does this trait, more than any other, gather all these stories, the IQ, the eyes, the immunity, the golden blood, the psychic gifts, the lost lineage, the sense of being from somewhere else? The honest answer has a name, or two. The first is rarity itself. When something about you is uncommon, the human mind reaches, almost helplessly, for it to mean something. You're rare and it's pure chance, just the lottery of which gene copies you happen to inherit. That's cold. It offers nothing. You're rare and it makes you special, marks you out, connects you to something hidden and grand. That's warm. That gives the rarity a purpose. And we are creatures who crave purpose so deeply that we will manufacture it from almost any raw material, including a missing protein on a blood cell. A rare blood type becomes a secret club, a private identity, an explanation at last for why you've always felt a little apart, a little different, a little like you didn't quite belong. That's not stupidity. It's one of the most human things there is, the hunger for a story that makes the strangeness of being yourself feel like it was meant. The second name is the Barnum effect, the well-studied tendency to read a vague, flattering general description and feel, powerfully, that it's about you specifically. You're sensitive, intuitive, you feel things deeply, you've always known you were different. That fits almost everyone, because almost everyone, in the privacy of their own head, feels sensitive and intuitive and a little apart. Attach that universal feeling to a rare blood type, and the blood type seems to explain the feeling, when really the feeling was there all along, in the negative and the positive alike, in all of us. And here's the cleanest proof that these are stories we hang on blood, not facts we read out of it. Travel east to Japan, and you'll find an entire popular culture built around blood type and personality, but not around the Roach factor at all. There, the system that's supposed to reveal your character is the ABO type, whether you're an A, a B, an O, or an AB. People discuss it the way others discuss star signs. It shows up in dating, in workplaces, in how people explain each other. Same human need, same biology underneath, and a completely different myth, attached to a completely different letter, in a completely different place. The West built its mythology on the RH factor. Japan built its on ABO. If the meaning were truly in the molecule, everyone would have landed on the same molecule. They didn't. Which tells you, as plainly as anything can, that the meaning was never in the blood. We brought the meaning, we always do. The molecule is just the hook we hang it on. None of which makes the real thing any less extraordinary. It just means the wonder is in the true story, where it belongs. And the true story, as we've seen, has plenty to spare. Strip away every myth then, the aliens and the crowns and the secret powers, and you might expect to be left with something deflating. A missing flag, a statistical curiosity, a riddle no one solved. But that's not what's left at all. What's left when you clear away the inventions is something real and quietly remarkable that the inventions were actually obscuring. So let's end with what this blood genuinely does, in the world, for actual people, because it's better than the fantasy. Start with the moment of crisis. A trauma patient arrives, bleeding badly, unconscious, with no time to do anything but act. There's no time to test their blood type or cross-match it, no time for anything but the fastest possible decision. What do the doctors reach for? The blood least likely to trigger a catastrophic reaction in anyone, the one stripped of all three of the major flags. It carries neither the A nor the B marking, and crucially it lacks the D flag too, which makes it the type the widest range of bodies will accept without their immune system screaming. That's O negative, the universal donor. In those first desperate minutes, before anyone knows who this person is or what runs in their veins, O negative is the blood that buys time. It is, in the most literal sense, the blood that strangers are kept alive on. The slogan oversimplifies, and since we've been honest all night, let's stay honest here too. O negative isn't truly perfectly universal. Blood carries dozens of other antigen systems beyond ABO and RH, systems with names like Kell and Duffy and Kidd and MNS, and in a small number of cases those can matter too. So universal donor is a useful emergency approximation, not an absolute law. And because O negative is both the safest default and relatively uncommon, it is almost always in short supply. Blood banks live in a state of chronic low-grade worry about running out of exactly this type. Which means there's a real and unglamorous truth here. If you carry it, your blood is disproportionately, concretely useful in the worst moments of other people's lives. The same rarity that made it seem mysterious is the rarity that makes it precious in a refrigerator in a hospital. Not a superpower. Something better than a superpower, because it's real. A thing you can actually give that can actually save a stranger. And then there's the frontier, where this old, strange blood meets the future. For decades, the dream in transfusion medicine has been to get past the shortages and the matching problems entirely, to manufacture blood, to grow red cells in the laboratory, or build synthetic ones, engineered deliberately to lack the troublesome flags so they can be given to almost anyone, produced in batches, never running out, never depending on whether enough of the right donors showed up that week. This is not science fiction. It's active research, with the first cautious human trials of lab-grown red cells already underway. And here is the lovely closing of the circle. To build blood that's safe for everyone, scientists need to understand the rare blood types deeply, including RhD negative, including the golden Rh null blood we met earlier. The very blood that evolution kept trying to delete, the absence that should have been a dead end, turns out to be one of the reference points from which we may someday write the recipe for the blood of the future. The thing that nearly disappeared may help make sure no one ever goes without, And the deepest real meaning, if you want one to carry off into sleep, isn't a power hidden in your cells. It's something quieter, and I think more true. Simply by being here, by carrying this blood at all, whatever type it is, you're carrying forward a thread that survived everything. Think of what it took. That stretch of code, present or absent, came down to you through an unbroken chain of people who each, every one of them, lived long enough and was lucky enough to pass it on. Through ice ages, through famines and plagues that emptied whole regions, through the brutal arithmetic of childbirth in eras when a mother and child surviving was never to be assumed. Every single one of your ancestors, all the way back to that person in Africa in whom the gene first slipped away, made it through. Not most of them, all of them, because if even one had failed, you wouldn't be here to wonder about it. You are the living end of an unbroken line of survivors hundreds of thousands of years long. That's true whether your blood is positive or negative. The RHD difference is just which note you happen to carry in a very old, very long song that has never once stopped playing. So let's come back at the end to where we started. Right now, as you lie here, roughly 1 in 7 people alive is carrying it. A small absence, a missing piece of DNA on the first chromosome, a single flag that most of humanity flies and they do not. By every rule that the living world runs on, it should have been erased long ago. It can, under the cruelest circumstances, turn a mother's own body against the child she carries. It cost untold generations their later children, invisibly, for longer than we have had history to record it in. It should have bled to nothing, should have become a forgotten genetic footnote. Instead, it rode out of Africa in the blood of our ancestors, spread unevenly across the whole world, and climbed in one quiet mountain corner of Europe, among a people whose language has no relatives on Earth, to the highest level found anywhere on the planet. About 1/3 of the people there, and nearly half of all the copies of the gene, and after everything we can now do with it, mapping exactly where it sits, reading the precise stretch of missing code letter by letter, even preventing with a single injection the harm it caused for 10,000 years, we still cannot say for certain why this blood is here. After 90 years, with every genome sequenced and every statistic sharpened, and every wild theory cleared away, the riddle, the genuine one, is still open. It is open tonight. It will be open tomorrow. Someday, maybe in your lifetime, someone may close it, but not yet, and there is something I find almost comforting in that. That after all our cleverness, the human family still carries, in its own blood, a question it cannot answer about itself. And now, the small thing I asked you to hold on to right at the beginning, the promise I said I'd keep. I asked you in the dark to wonder whether you might be one of them, one of the people carrying this blood, one of the others resting somewhere out there with the same small absence in their veins. Here is the easy, quiet way to find them if you'd like to. Somewhere down below this you can leave a word, where in the world you're listening from, and what the time is there, in your room, right now. And if you happen to know it, only if you'd like to share it, your blood type. You don't have to. But if you do, something quietly lovely happens. The map we spent tonight tracing across the whole human species starts to fill in, not with statistics, but with real people. One of them in Tokyo, where it's already getting light. Another in a kitchen in Ireland near midnight, the kettle just gone quiet. Another in the Basque Mountains, where the whole story reaches its peak, where it runs higher than anywhere else alive. The same blood. The same open question. The scattered others, who spent tonight alone in their own dark rooms, made visible to one another for a moment. A constellation of people who, by every rule, weren't supposed to still be here, finding each other in the comments under a story about why they are. There is no need for it to be alien, no need for it to be royal or magical or a sign of anything but what it is. The honest version is enough, and it goes like this. Somewhere in the deep past, in a single person whose name and face are gone forever, a small piece of genetic code went missing. It should have been a dead end. It should have stopped there. Instead, it was passed down to that person's children and to their children, and onward through everyone who survived long enough to carry it one generation further, an unbroken chain across hundreds of thousands of years, all the way down to you, or to someone you love, or to a stranger resting in the dark on the far side of the world tonight with the same question in their blood. It's still there, in 1 in 7 of us, moving quietly through the dark, doing its old and half-understood work, holding its secret close. We don't know why it stayed. Maybe one day we will. For now, you don't need the answer. You can simply rest inside the question. Good night.